Okay, let's unpack a really profound medical paradox today. We're talking about tamoxifen with it. I mean, a true hero drug, really, since the 1970s. It's credited with saving millions of lives, in the fight against breast cancer. A real triumph, offered so much hope where honestly there wasn't much before. But, and there is a but, this life-saving medication carries this rare kind of perplexing secret. For decades, doctors have seen this link, never fully understood it though, between tamoxifen and a slightly higher chance of uterine tumors. So our mission, let's say, in this deep dive is to finally get grips with the why, the specific molecular mechanism behind this puzzle. And we're drawing on some really recent breakthrough international research.
Yeah, all this insight comes from a great report in SciTech Daily. It breaks down findings published in Nature Genetics, a top journal. And the research wasn't just one group. It was this big international team effort. You've got folks from the Berlin Institute of Health at Charité, the Broad Institute of MIT and Harvard, Mass General Brigham, Dana-Farber. I mean, really top minds working together.
Shows what happens when science collaborates globally, right, on these critical health questions. So maybe first, let's just take a second to really appreciate tamoxifen's impact because, I mean, since the 70s, it's been absolutely central, hasn't it, to improving survival for millions with estrogen receptor-positive breast cancer. Its effectiveness is just, it's undeniable, a cornerstone.
Oh, completely. What's fascinating here is just the sheer scale of its success, how fundamental it became worldwide. For a long time, we've understood that many breast cancers are fueled by hormones, specifically estrogen. Tamoxifen is what we call a SERM, a selective estrogen receptor modulator. Basically, it sits on the estrogen receptors on those cancer cells, blocks the estrogen from getting in and telling the cell to grow. So it cuts off the fuel supply, essentially, stops the cancer progressing. That mechanism made it incredibly crucial, a game changer, quite literally, for so many women.
And it still is, giving people years, decades, more healthy life. But like you said, and like we started with, there's always that little asterisk, that but. This known, though infrequent, higher likelihood of uterine cancer. It's just been this nagging question mark hanging over this amazing success story. That's the paradox. And here's where it gets really interesting, right? Because for decades, the reason why tamoxifen was linked to uterine cancer at a cell level that was a mystery. Doctors knew it happened sometimes, but the underlying biology, unclear. Expert, this new research, it sounds like they finally cracked it. What exactly did they find?
They absolutely have. It's quite elegant, actually. The core finding is that tamoxifen directly stimulates a major cellular signaling pathway. It's called PI3K. Now, for anyone familiar with cell biology, the PI3K pathway is like a master switch for cell growth, survival, proliferation. Think of it like an internal communication line in the cell telling it what to do. And PI3K is critical because when it gets stuck in the on position, it's a known driver in lots of cancers, including, importantly here, spontaneous uterine cancers, the ones that just pop up without an obvious external trigger. It's a key growth pathway.
Okay, so that really highlights the paradox then. Because wait, if PIK3CA gene mutations are the usual suspects for turning on that PI3K pathway in those spontaneous uterine tumors, how did they figure out tamoxifen was doing it directly? Especially when, as the report says, those PIK3CA mutations are actually less common in the tamoxifen-linked uterine cancers. That seems backwards somehow.
You've hit the nail on the head. That's the conundrum they solved. What the researchers discovered, essentially, is that tamoxifen itself makes the mutation unnecessary. So instead of needing that genetic glitch, that PIK3CA mutation, to flip the PI3K switch, the drug itself provides the on signal directly. It's like tamoxifen walks in and says, don't worry about the faulty wiring, I'll just turn the lights on myself. And by doing that in uterine tissue, it unfortunately drives tumor development. It's really quite a remarkable finding. Professor Kirsten Kubler, one of the lead researchers, put it very clearly. She said, our results show for the first time that the activation of a pro-tumor signaling pathway by a drug is possible and provides a molecular-level explanation for how a highly successful cancer drug can paradoxically promote tumor development in another tissue.
Wow. Activation of a pro-tumor signaling pathway by a drug. That's a heavy statement. Makes you think differently about how drugs work, doesn't it?
It really does. And she added, tamoxifen bypasses the need for genetic mutations in the PI3K signaling pathway by directly providing the stimulus for tumor formation. So if we connect this to the bigger picture, like you said, this really shifts our understanding. We used to think mostly about therapy-related cancers as The drug causing mutations, damaging DNA, which then leads to cancer. This research challenges that. It shows a drug's direct action, not necessarily causing a mutation, can substitute for those genetic changes. It can directly activate the same cancer-promoting pathway. It's a whole new mechanism to consider for drug side effects.
Substitute for genetic changes.
Yeah.
Yeah, that feels like a real paradigm shift.
Yeah.
Does this mean we need to maybe reevaluate how we look for drug side effects more broadly?
Mm-hmm.
I mean, if a drug can just switch on a growth pathway directly without a mutation, what else might be happening with other drugs?
That's exactly where this leads. It's the exciting sort of next step. It tells us we need to look beyond just DNA damage or mutations. When we assess drug safety, we need to consider direct pathway activation too. For pharmacology, yeah, it suggests a more holistic approach maybe to drug development, to monitoring,
Right.
looking at the whole signaling network a drug might affect, not just its main target. You can imagine new kinds of screening tests, perhaps, looking for this kind of direct activation of oncogenic pathways.
That really does sound like a leap forward. Now, okay, just to be super clear, because this might sound alarming, it's really crucial to stress that the overall risk, the actual risk of getting uterine cancer while on tamoxifen is still very low. We're talking about a rare side effect here, even though it's serious when it happens. This isn't panic stations.
Absolutely not. That's critical context. The life-saving benefits of tamoxifen preventing breast cancer coming back, treating it, these far, far outweigh this rare risk for the vast majority of patients. This research isn't about saying tamoxifen is bad. Not at all. It's about understanding it better, understanding exactly how it works, the good and the rare bad at a really detailed molecular level. It's science pushing forward to make a vital medicine even safer, potentially more personalized down the line.
Right, and that's the positive spin, isn't it? This discovery, while it explains something complex and unexpected, it actually opens doors to making treatment even safer for breast cancer patients. It's not just an explanation, it's a signpost for improvement.
Precisely. Having this clear biological reason for this old puzzle, it lays the groundwork for more personalized ways to prevent or manage this side effect. You can imagine, maybe in the future, we could identify patients who might be at slightly higher risk. Maybe through biomarkers, maybe their specific genetic profile related to PI3K sensitivity, something like that. And for those patients, you could maybe think about strategies to mitigate the risk, perhaps combining tamoxifen with something that specifically dials down PI3K in the uterus, or adjusting the dose, or maybe using alternative drugs in that class. It opens up possibilities for real precision medicine, making a life-saving drug fit the individual better. Which raises that important question, doesn't it? How do we use this insight to make this drug even safer for everyone?
That's the goal, isn't it? Tailoring treatment. And it sounds like the researchers are already thinking bigger picture, too. They mentioned plans to look into whether similar mechanisms, this direct pathway activation, might be involved in side effects of other drugs, which hints at, well, much broader implications across medicine, potentially uncovering hidden effects of many common treatments.
Exactly. This could be a really fundamental shift in how we approach drug safety and development. It's basically a call to look much deeper at the off-target effects. Not just what pathways a drug blocks, but what it might accidentally switch on. Think about it, thousands of drugs out there. How many might be subtly tweaking pathways and tissues we haven't considered, maybe causing low-level, long-term side effects we haven't connected the dots on yet molecularly? It's a huge area to explore.
So, okay, let's bring it back. What does this all mean for you, listening in? The key takeaway. Tamoxifen, this vital breast cancer drug, can directly activate a key cancer pathway, PI3K, in the uterus. This activation kind of substitutes for the usual genetic mutations, and that explains its rare link to uterine cancer.
And beyond just tamoxifen, this whole deep dive really highlights the process of science, doesn't it? Taking something doctors observed for years, the side effect puzzle, and finally digging down to find the precise molecular why. It just reminds us that really understanding how drugs and our bodies interact, right down at the cellular level, that's what leads to safer, better, more personalized medicine for all of us. It's about constantly peeling back those layers of complexity, refining our tools.
Absolutely. And maybe the provocative thought to leave you with is this. Just consider how many other drugs we use every day might have these undiscovered molecular actions, affecting different parts of the body in ways we just don't know about yet. What other breakthroughs, what new ways to make treatment safer or more targeted might be waiting just around the corner when scientists start looking for these mechanisms, like they plan to here? This work is a great reminder that even our most trusted medicines, ones we've relied on for ages, still hold secrets. And uncovering them pushes our understanding forward, ultimately getting us closer to truly precise therapies.